had that beginning of life. All right. So biogenesis, of course, was the first part of our lecture today, and the other day. And keep getting my feet caught in my mouse cord. I don't want to end up tripping myself. So what we discussed in that was the whole idea of how life began on Earth. And of course, I gave you 4 very broad points about the way that worked. Nobody's saying that's exactly the way it happened, but, it's one of those things where we were, as scientists, we're saying, if life began on Earth, then these are the conditions we feel based on what we know about Earth. These are the conditions we think that would have been necessary. These are the events that we think would have been necessary in order to bring that life to occur. So we've gone over that. Does anybody have any questions on that before I actually get on to the material today? Does anybody have any questions about the biogenesis stuff that we're looking at, the idea of the protocells and all that stuff? It's all kind of interesting and it's fascinating to think about, just how things like that come together. We're all good? All right, so let's get started here. So again, starting back to the beginning, the Earth is the solar system and the Earth about 4.5 billion years. It was about 1 billion years later when life shows up. Again, I've talked to you that there are some thought out there that life may have started and ended a couple of times between when the solar system or when the Earth came into being, and we actually see evidence of it. But life that left behind some sign. Now, there weren't any clear fossils before about 3.8 billion years ago. And again, that doesn't mean that life didn't start before that. But fossils, as we've talked about, are very difficult to find. And we're talking about fossils of large organisms, not to mention some of the early critters on the planet, which would have presumably been, small little single-celled critters. And they're soft-bodied organisms again too, and things like that just don't fossilize as well as things that obviously have bones. So that 3.8 billion years is probably not a bad assumption, especially what we know about the Earth and and what it was like at the beginning, and how those conditions weren't, while being conducive to making complex molecules, they certainly weren't conducive to life as we know it. The first thing we'll start off with today is we'll move on and talk about the prokaryotes. And of course, we talk a whole chapter about those, but we need to discuss them in the context of life on Earth. And among those prokaryotes, there were some of those prokaryotes that were actually engaging in photosynthesis. So this next thing that happened somewhere around 2.75 billion years ago, this rise of atmospheric oxygen was extremely important because that changed the world considerably. What we find many times with organisms is the fact that sometimes organisms will actually create an environment that's more conducive for later organisms to move out onto. Like today, we're going to talk about a little bit about the colonization of land, when organisms begin to move out on the land. And if you think about it, in a very basic sense, the first critters that moved out on the land had to have been the plants, right? Had to have been the photosynthesizers, because the animals, there was nothing to eat out on land. So that's what I mean by the fact that sometimes organisms will actually change the environment or make the environment more conducive for other critters to survive. So this whole idea of this rise of atmospheric oxygen is important because that changes a lot about how organisms go through this the process of respiration that we learned about. So we'll come back to that, obviously. With the rise of atmospheric oxygen, complexity could increase. So we started getting eukaryotic cells, and initially they were single cell for quite a period of time, and then they began to be multicellular. But for 1 billion years or so, we were pretty much limited to relatively simple eukaryotic organisms, very, very simple type of animals, very ancestral, like sponges and jellyfish. And again, those critters are incredible and sophisticated in their own right, but in the overall scheme of things, those were we would consider the more ancestral types of organisms. So for about 1 billion years, those were the organisms that pretty much had a place to themselves. And just about where you see the animals starting, just about that time when we talked about the Cambrian explosion. At the Cambrian explosion, we get this rapid diversification of life. Now, it was all life, but animals are one of the ones that saw the biggest diversification of life. We went from just a few simple animals before and after the Cambrian explosion, exploded into most all of the common groups of animals that we have on Earth today. And soon after that was the colonization of land. And then here we come again at the very end of it all, that little tiny sliver, and us humans trying to figure out what was happening for all these billions of years before that. So this is not any easy task. So let's step back again and review one more time about what the early Earth was like. So about 3.8 billion years ago, the Earth was not a very nice place. It was very violent. There was a lot of asteroids. As I mentioned to you, the solar system was it was like a pinball gallery. There was just rocks and stuff flying all over the place, and bombardment of planetary bodies. And in the early Earth, there wasn't any water because of the heat from all this bombardment. And when these rocks and these big, planet-sized rocks crash into one another, they release a lot of heat. And that heat wouldn't allow the water to stay liquid. Obviously, it would boil it off. Now, water then began to collect on Earth. One of the theories I think I mentioned you guys with water perhaps got to Earth by being delivered by comets. Comets are, they call them dirty snowballs. Basically, comets are ice and snow. Makes you wonder how many comets must have ran into the Earth to be able to dump enough water based on what we see today. But just about that 3.8, 3.9 billion year ago, somewhere in that vicinity, the Earth begins to cool off and liquid water starts to form. So now we got conditions that would be a little more conducive for life. Nothing really too fancy, though. We're going to start off, of course, with the prokaryotes, right? The members of the domain archaea and bacteria. And these two groups of organisms, they are the king of the Earth's history, really. They were around long before we were around. Presumably, they're going to be around a lot longer after we're gone. 3/4 of the Earth's history is made up of these prokaryotes. It wasn't too long, though, after life arose that these two groups of organisms broke off into these two domains. Now, you remember from Bio 1 talking about domains. There are 3 domains. There's archaea, bacteria, and then what was the third domain? What domain are we in? Yeah, eukarya. Thank you, Harrison. We're in the domain eukarya. So what I want you to understand about that is very early in the Earth's history, the prokaryotes broke off into these 2 separate domains, which means they're substantially different from one another. Again, if you think about it, we're in the 3rd domain. So the amount of difference, or the difference between us and the members of the domain bacteria and members of the domain archaea is the same as the differences, more or less, between the archaeans and the bacteria. It's something we tend to do in human nature sometimes. We kind of lump all of the prokaryotes together. As a matter of fact, when I took biology, when I first started teaching this class, to be honest with you, we didn't have the domain system. We had kingdoms, of course, and all of the prokaryotes were in one kingdom, the kingdom Monera. Now, there no longer is a kingdom Monera because we found out that putting those organisms, those 2 groups of prokaryotes into the same kingdom was actually very artificial. They're very different from one another. The archaean prokaryotes, and the name kind of tells you what it is, they're the ancient ones, the archaic ones. And many times in the archaeans, we have a lot of the very extreme prokaryotes that I was talking about the other day when we went through the prokaryotes. They can live in all kinds of very extreme conditions. There are a lot of bacteria that can do that too, but the archaeans are well known for being extremophiles. So even though the early Earth may not have been great for us when it comes to life, these organisms that were able to deal with such harsh conditions like heat, salinity, toxic fumes and all those, and radiation, because remember the early Earth, there wasn't any oxygen. So if there wasn't any oxygen, there wasn't any ozone. And if there wasn't any ozone, there was nothing filtering out the ultraviolet light. So these early critters had to deal with high levels of radiation, of UV radiation, which can cause all kinds of genetic mutations. Although, as we mentioned the other day in the prokaryotes, that might have been a bad thing for them, right? That might not have been a bad thing to be in an environment where there's a lot of genetic mutations going on, because remember, in the prokaryotes, they can get away with genetic mutations. They can get away with genetic mutations in the sense of, even though they may be as detrimental, as we've talked about, whenever you monkey with the genes, you have to, really sort of taking a chance. But the amount of genetic variation and change that was occurring from the UV, even though it may have been detrimental to a lot of these prokaryotes, the other day we were discussing the fact that since they reproduce so fast and reproduce so plentiful, that, you know, you could get away with that. If one in a billion genetic mutations was beneficial, well, E. coli and other prokaryotes can crank out a billion offspring pretty doggone quick. So you can play the odds game on that. So we break off into these two groups of prokaryotes, relatively simple organisms, but like any living organism, no matter if you're an archaea, bacteria, an animal, or a plant, you have to have some way of converting energy, of obtaining energy from your environment. You need energy to carry out all those chemical reactions, those very specialized chemical reactions that we talked about in biogenesis. So some of the first prokaryotes were more than likely chemoautotrophs. Now, autotroph, of course, is you make your own food, and in chemoautotroph, you use chemical reactions by which to do that. Compare that with the word down below, the photoautotrophs. Photoautotrophs are, of course, the organisms that feed themselves and use light as the form of energy, right? So these would be, for the most part, your plants, your photosynthetic critters. But again, some of the earliest prokaryotes may have been chemoautotrophs, and perhaps down around those geothermal vents that we were talking about. Maybe it wasn't exactly like we talked about with the Urey-Miller experiment, but down around those geothermal vents where there was a lot of carbon dioxide and hydrogen, a lot of sulfur, a lot of hydrogen sulfide, organisms could have been around and are still around today, as a matter of fact, who can actually engage in this process of creating food by doing something like taking carbon dioxide and hydrogen and combining them together to make methane. These critters are actually called the methanogens. Methanogens derive energy from that chemical reaction. The oxidation of sulfur, the oxidation of hydrogen sulfide, or even the oxidation of iron, like this critter that I mentioned the other day in the picture there. This is the bacteria that was discovered on the Titanic, Monas titancea, I believe. I think that's, I don't know if that's spelled exactly right. But this was a fascinating find for microbiologists, because what we discovered about this critter is it's a chemoautotroph. It obviously isn't a photoautotroph. The Titanic sank in about 2 miles of water, so there's not any sunlight down there. But they actually oxidized iron. They actually oxidized the iron in the plates. Now, you may not remember, you know, the relationship between energy and oxidation and reduction. You talked about it in Bio 1. You remember reduction is the gaining of electrons, and the oxidation is the loss of electrons. But there was the connection was made later on in the chapters when we were talking about thermodynamics and enzymes and their part. When you reduce something, when you add an electron to something, you're building something larger. And when you oxidize something, you're taking an electron away from it, so you're tearing apart. Now, the laws of thermodynamics tell you that when you build something, when you make something, when you take little things and make them and put them together, you need energy. And when you take that big thing and break it apart, the energy is released. So that's what these chemoautotrophs were doing with sulfur or hydrogen sulfide, or in that really bizarre case, iron. They're oxidizing those materials, and the oxidation is releasing the energy that they need to carry out their metabolic processes. This obviously, you know, wouldn't work for us, but we still do engage in oxidation, right? That's what's going on during respiration. Remember, respiration electrons are being torn off of materials and then passed through a proton pump to pump protons. That energy that's being torn off of those, that's being released when you oxidize those materials, is some of the energy that's necessary for respiration. But for those chemoautotrophs, that was fine. But finding those areas on Earth, and even back then there was a lot of hydrogen sulfide, but as time went on, finding those areas on Earth where you would find these types of chemicals you could oxidize probably got rarer and rarer with time. But one thing that there has always been a lot of on Earth is sunlight. Yeah, we're only 93 million miles away from the sun. So a lot of critters out there became photoautotrophs, and they used sunlight to engage in that process of converting energy, of taking sunlight energy and converting it into food energy. Now, I want you to take a look at these two cladograms, just like the cladograms we did in class the other day. And remember, the important thing about a cladogram is the Occam's razor idea, or the idea of parsimony, the simplest one. Now, remember back to Bio 1 when you talked about photosynthesis and respiration. photosynthesis was a pretty complex set of chemical reactions, wasn't it? I mean, there's the light reactions and the dark reactions, and there's the proton pumps. I mean, so that's a fairly sophisticated metabolic process. So taking a look at these two cladograms, the one on the left says that some ancestral prokaryote initially arose and wasn't able to photosynthesize. And as time went on, there was, what do you got, 5 in this example, 5 lineages that broke off that developed, independent of one another, the ability to photosynthesize. The alternative is the one on the right. In this case, the ancestral prokaryote could photosynthesize, and the ones that become heterotrophs, they just basically lost the ability to photosynthesize. So now apply that parsimony principle to that again. Which one of these two do you think is most likely? The one on the left, where photosynthesis evolved, several times in different lineages, or the one on the right, where photosynthesis evolved once, and if you're heterotrophic, you just basically lost the ability to photosynthesis? Which one do you think is probably the most parsimonious, or which one is maybe the least probable? So you think the right is the best, is probably the better word, or is the, I just totally tripped over my tongue. The one on the right is yeah, most likely the one on the right, right? Yeah, because photosynthesis, like I said, is a pretty complex process, and the chances of that complex process evolving more than once, although not impossible, right, is not the most parsimonious. The most parsimonious, and other evidence supports this, was that the ancestral prokaryote, right, Tiffany, that's exactly right, it is the simplest, evolved, so photosynthesis evolved once, all right? Now, it's been modified through time, don't get me wrong, but the basics of photosynthesis evolved once, and then other creatures just lost the ability to photosynthesize with time. Okay, photosynthesizers, what gas do they produce? Well, I shouldn't say produce. Yeah, what gas do they release? What gas do photosynthesizers release? Yeah, that's right, it's oxygen. Okay, and you remember the early Earth didn't have any oxygen, or had very little oxygen. And if you remember from photosynthesis, the oxygen comes simply from the splitting of water, right? I mean, water for a plant, in terms of photosynthesis anyway, is just a source of protons and electrons, right? You need the protons from the hydrogen, you need the electrons from the hydrogen, so you strip that stuff off of water, and then the oxygen is given off as a gas. So, when we say plants create oxygen, I like to think, well, they just basically strip everything off of water, leaving behind the oxygen. All right, but that oxygen isn't being produced by these organisms, for any purpose. There's no respiration at this particular time. So that oxygen is essentially kind of a waste product, if you think about it. But here's where I go back to where I said at the beginning about organisms changing the environment for other organisms later on down the road. Now, they don't do this altruistically, folks. They don't do it with thinking, I'm going to make it better for future generations. That's not the way it works. This is just sort of a happenstance thing that occurs. So as the oxygen levels begin to rise in the atmosphere, as the oxygen levels begin to be, or as oxygen begin to be produced by these organisms, most of these organisms were aquatic, so the oxygen levels begin to rise in the water. That's the first place. The cyanobacteria, these are the critters I'm talking about. Like, for example, Anabina and Oscillatoria that I showed you the other day, those are cyanobacteria. And cyanobacteria photosynthesize, and they produce oxygen. And there were massive mats of these things, folks, just huge sheets of Anabina and Oscillatoria growing in these huge, massive expanses. What's a stromatolite? Down in the lower right are pictures of stromatolites. I think these are the ones from Shark Bay in Australia, I believe. Let's see. You may not have gotten that picture out of your book, but I think this is from Australia. But what you're looking at here is the mineral deposits that were left behind by these ancient cyanobacteria. And it looks like they're in, lumps, if you will, but basically the way this occurs is this was one huge fossilized mat or fossilized structure of these cyanobacteria, and then the ocean, which you can kind of see in the background, moves in and erodes, and that's what causes all these pillars to form. But at one time, this would have been just one giant collection of these cyanobacteria. What I'm trying to get across here, folks, is the number. We're talking about a bunch of cyanobacteria producing a bunch of oxygen. Now, again, they're aquatic, so first the oxygen stays in the water and combines with the iron, so it makes rust, right? Iron oxide. But as the oxygen levels begin to rise in the atmosphere, or in the water, eventually that's spilled over into the atmosphere. So we started getting a rise in atmospheric oxygen. Now, atmospheric oxygen, its rise was good for some critters, but not so good for others. For example, there were many anaerobic prokaryotes out there that couldn't deal with the oxygen. Oxygen is a heavy oxidizer, so it was destroying these anaerobic prokaryotes. We even have some of these prokaryotes alive today. They're called facultative, obligate anaerobes. Obligate anaerobes are creatures that die, and they're prokaryotes, folks. Prokaryotes that die in the presence of oxygen. I'm sorry, I got that backwards. It's facultative are organisms that can do aerobic or anaerobic respiration. But if you're an obligate anaerobe, you die in the presence of oxygen. So a lot of these early anaerobic prokaryotes were obligate anaerobes. In other words, oxygen was destroying them. Here are a couple of slides. I'm going to show you, though, that someone came up with this really clever solution involving a symbiotic relationship. We see that a lot in biology. Organisms that are struggling to survive will sometimes get an advantage if they enter into some sort of a symbiotic relationship with another organism. So I'll talk about that in just a second. But about 2.2 billion years ago, we have this incredible rise in oxygen. And this is when a lot of the eukaryotic organisms started coming on the scene, eukaryotic algae. Let me say something about algae for just a second before we go on. The word algae means many different kinds of organisms. For example, there are prokaryotic algae. They're the like Anabina. They're what we call the blue-green algae. So blue-green algae refers to prokaryotic organisms. There are protozoan algae. For example, seaweed. Seaweed is not a plant. Seaweed is actually a protozoan. And sometimes we collectively call species of seaweed like sargassum and kelp, we call it brown algae. So algae can be referred to a protozoan. And then there's green algae, which is a plant. So the word algae can refer to either a prokaryote, a protozoan, or to a plant. In this particular case, we're talking about the rise of eukaryotic algae, whether they be protozoan or plant-like, along with chloroplasts. And this coincides very closely with that rise in atmospheric oxygen. Now, how do we know that that rise of atmospheric oxygen occurred at about that time? Well, you remember I told you that we can take little samples out of like glaciers or rocks, little bubbles, and sample the atmosphere. And when they've done that, look at that chart that I've got there. They noticed that about 2.5 billion years ago, we had something called the oxygen revolution. Look at the oxygen levels went up dramatically. All right, and when those oxygen levels went up dramatically, that's when that coincides with the rise of eukaryotic life. Now, think for just a second. Why would the rise of oxygen be beneficial for eukaryotic life? Why would the rise of oxygen be important for more complicated life? Think back to respiration. Specifically, think back to respiration and the amount of ATPs that you get from respiration. How many ATPs do you get from one glucose if you run through the entire process of respiration? Yeah, it's the final electron acceptor. That's right. Oxygen is. And that allows those electrons to go through the entire, the glycolysis, the Krebs cycle, then through the electron transport chain in the mitochondria. But how many ATPs do you get out of that on average? One glucose will yield how many ATPs? Nobody remembers? Yeah, yes, 36 to 38. Yeah, 36 to 38. I usually just kind of remember the 40 because it's a nice round number. It's not exactly every time because nothing's 100% efficient. Okay, how many ATPs do you get from anaerobic respiration? Now, in anaerobic respiration, you only do glycolysis, remember. So how many ATPs do you get out of that? Can't hold my hands in front of me. No? 6. It's not very many, but less than that. 2, right? You get 2 ATPs. Remember, you actually get 4, but you use 2. So we say it's a net yield of 2. So now, thinking back what we've been talking about during the evolution chapters, which one of those organisms would have been more efficient? The one that's the eukaryotic, which is getting 36, 38 to 40, or the one that's the prokaryotic, especially if it's an obligate anaerobe, you can only get 2. Which one's more efficient? Yep, eukaryotic one, right? Now, when you're more efficient, that means exactly what it says. You make more efficient use of that energy, so therefore you can increase in complexity. Prokaryotes were limping along, they still are today, limping along, getting 2 ATPs if they're anaerobic, out of a molecule of glucose. But along comes this other life type, this eukaryotic life type, that can use these rising oxygen levels and take that glucose and not get 2 ATPs, but get 38 or 40 ATPs. You're talking 16, 17, 18, 20 times more energy from the same glucose. That's going to drive efficiency. That's going to be efficient. That's going to drive diversity. That's going to drive complexity. So these prokaryotic organisms that were photosynthesizing were unwittingly setting the stage for more complex life. And the reason why the oxygen was the key behind that was because with oxygen, you can extract more energy out of a glucose. And if you can extract more energy, you can do more things with it. So you can become more complicated, okay? That make sense? All right, as I said, a lot of the anaerobic prokaryotes, they were on the run, and some of them actually went extinct as these oxygen levels began to rise. But as I mentioned, some survived through endosymbiosis, which I'll show you an example of that in just a minute. So let's take a look. We're jumping up now just a little over 1 billion years. Fossils from 1.8 billion years ago show us what might have been multicellular eukaryotes, but the oldest known confirmed, if you will, is from about 1.2 billion years ago. Remember, don't be taken aback by that, such a substantial difference in time. Remember what we've talked about with fossils. That explains that large gap. Plus, you're trying to estimate things in 1 billion years, so if you're off by 100,000 years or so, it's really not that big of a deal. So this oxygen level begins to rise, okay? That allows for greater complexity. And greater complexity means that eukaryotic or life forms can have organelles, and that's division of labor, right? Golgi apparatus, endoplasmic reticulum, the mitochondria, the chloroplast, all those things we learned about. Now, prokaryotes don't have that. They got ribosomes, but that's about it as far as organelles go. And another thing that this greater complexity allowed eukaryotic cells to do was with the advent of the cytoskeleton. Remember the microtubules and the intermediate filaments and the actin filaments and all that stuff that you learned about in Bio 1, which allows the cell to stay together and keep its shape, allows it to move. Another thing that the cytoskeleton allows cells to do, or provides the ability for the cells to do, is engage in what you see in the picture down below, A through D. What's going on in A through D? All right, so the right-hand side of each picture, that's going to be the inside of the cell. There's the inside, and this is the outside of the cell. So what are we witnessing as we move from A to D? What process is that? What is that cell doing? A secretory vesicle, Tiffany, would be one that would be going the opposite way, right? A secretory vesicle would be a round vesicle that the cell actually sends into the environment, that creates and sends into the environment. This is the opposite of that. Yeah, there you go. Endocytosis, phagocytosis if it's something large, pinocytosis if it's a smaller or a liquid molecule, right? So endocytosis is the engulfing of things, and that's what you see in the picture, right? We're engulfing something, aren't we? Now, prokaryotic cells don't do this. Eukaryotic cells are well known to be engulfers. So keeping that in mind, let's talk a little bit about this whole endosymbiosis for some of those prokaryotes that were being damaged by the rising oxygen levels. Let's take a look at a scenario. So in the upper left, we have some ancestral prokaryote that began to develop the ability to engulf. All right, and what it basically started out doing was it began to fold its membranes in on itself. And we saw that in the prokaryote the other day when we were looking at the the aerobic run and the photosynthetic run. I showed you the membranes. Remember I said all you need for very simple photosynthesis or respiration is a membrane. So these organisms, even prokaryotes, were already infolding the membrane, these invaginations that you see. And eventually what we have in a eukaryotic situation is those infolded membranes would surround the nucleus or the DNA so that we get a nucleus. And other things that they would have engulfed in addition to food, which you can imagine phagocytosis is exactly what that is, they could have been engulfed an aerobic bacteria, maybe similar to the one you see down at the lower left. And they could have engulfed and or a photosynthetic bacterium. Sort of like what you see in the upper right, that green mat there is a bunch of photosynthetic bacterium growing. Remember, very often in biology, if it's green, it's probably photosynthesizes. So now, rather than engulf these two bacteria, rather than engulf and eat them, what happened was they engaged in an endosymbiotic relationship, right? So the aerobic bacterium, through time, eventually becomes the mitochondria, and the photosynthetic bacterium through time eventually becomes a plastid. And a chloroplast is a plastid. A plastid is a type of organelle. So now we have a situation where this heterotrophic eukaryote, or this ancestral prokaryote, now has become a heterotrophic eukaryote or a photosynthetic eukaryote. So these things that were free living at one time, the photosynthetic bacterium and the aerobic bacterium, may have escaped the changing conditions on Earth by living or allowing themselves to be engulfed inside of this eukaryotic cell. And instead of being eaten, they entered into a relationship. Now, let's start with the photosynthetic one. What would have been the advantage? Okay, because this is what's going to drive evolution. So whenever we see these things happening, we always have to say, would evolution have supported that? What would be the advantage of a cell possessing a photosynthetic plastid or a chloroplast, as they are now called today? What advantage would that cell now have? What could it do that it couldn't do before? Well, yeah, produce oxygen, but you remember the oxygen production in photosynthesis is kind of a side note. What's the whole purpose behind photosynthesis? Why do plants today photosynthesize? Yeah, they make their own food, don't they? So now a cell that has a chloroplast or a plastid would have the ability to make its own food. So all it needs to worry about is the sun coming up. Now how about with the mitochondria? What would be the advantage of the mitochondria? I think Jared actually kind of hinted to it there. If you have a mitochondria inside the cell, you're not limited to just glycolysis, are you? You remember in Bio 1, glycolysis occurs in the cytoplasm of the cells. It's only when you have a mitochondria that you get the Krebs cycle and the electron transport chain. And it's only with those two things that you get the 40 or 36 to 38 ATPs, right? Without any of that, you only get 2. Now, those two things, yeah, heck yeah, it was. Those two things, the ability to make more food, and as Bradley says, with the mitochondria, you're much more efficient, that would be driven by evolution, wouldn't it? That's what evolution is all about, making organisms efficient. This would make an organism a lot more efficient. As a matter of fact, this is where we think perhaps the organelle mitochondria and the one chloroplast came from. I don't know if you remember from Bio 1, but mitochondria and chloroplasts, yeah, that's a symbiotic relationship. Mitochondria and chloroplasts both have their own DNA, and they both divide independent of the cell. So they almost act like a prokaryote. They have their own DNA And when they divide, just splitting, like binary fission, the way prokaryotes do. All right, so that's the endosymbiotic theory of the origin of mitochondria and chloroplasts. That's correct, Bradley. Mitochondrial DNA is only from your mother. The DNA in your mitochondria is maternal, completely maternal. All right, so now we've got this increasing complexity. So we're going to kind of fast forward a little bit, because through billions of years, complexity rose at a relatively regular pace. Animals were getting a little bigger, more multicellular. Critters were becoming a little more complex. And then along comes that thing that caused Darwin so much trouble. About 500 or so million years ago, in the fossil record, they went from very little organisms, just a few, again, the sponges, the jellyfish, and some of the other simpler critters out there, to this what seemed like an overnight appearance of all of the other major groups of organisms, including many of the major groups of animals, right? And this happened in the chordates. We're a member of the chordates. So the chordates arose very early on, just after during the Cambrian explosion. Now, Darwin had problems because it seemed like it went so fast to him. Remember, he didn't know about punctuated equilibrium. Remember, it was just gradualism with Darwin. So when he was aware of all these fossils that show up 500 million years ago in the fossil record, he was bothered by that because it seemed to occur pretty quick. It wasn't quite as quick as Darwin perceived. The explosion, the Cambrian explosion, really had a relatively long fuse. The Cambrian explosion lasted somewhere between 25 to 100 million years. So that wasn't exactly an overnight kind of thing, even in terms of geological time, right? Now, what was driving this explosion, however slow? Well, it was something called an arms race. You see, as we get up to the part of the Cambrian explosion, just about that time, what's happening is organisms are learning how to eat one another. And they're learning how, or not learning, but they're evolving structures enabled to do that. Grasping structures, the ability to swim after and attack and kill prey. So there became an arms race. Right? An arms race between the predator and the prey. So the predator becomes really good at exploiting and eating the prey. And if the prey doesn't respond, it could go extinct. So the prey may adapt and become a little better at getting away from the predator, or staying away from the predator, or somehow avoiding predation. And then the predator, of course, has to come back with something else, because if not, it could go extinct. So you see what I mean? It's an arms race. Each time one gains the advantage over the other, then it's necessary for that one that doesn't have the advantage to evolve or adapt in some way, so it now does have an advantage. So that's what we mean by that thing, the arms race back and forth. And whenever you have that kind of selective pressure, that actually causes adaptation to occur relatively quickly, right? That's a very heavy selective pressure, that arms race. So we think that was one of the big reasons why during the Cambrian explosion, we get this rapid proliferation of animal groups, because of the fact that there was a lot of speciation going on as these guys were dealing with these organisms were dealing with the arms race problem. It's like carnage back then. All right, now after the Cambrian explosion, just soon after, 500 or so million years ago, plants began to move out onto land. So, land plants began to, well, there were already plants in the water, but plants began to move out onto land. And like I said before, moving out onto land, plants actually made the land environment more conducive for animals. But let's step back for a second, and talk about what helped the plants. Plants, of course, they need sunlight and they need water, but they also need minerals, nutrients from the soil. And the soil back then didn't have a lot of these nutrients that plants needed. So plants moving onto land were faced with a dilemma. Moving out onto land might have been good because you might be escaping competition in the water or something like that, or predation by a herbivore, which is predation, if you think about it, if you're a plant. But moving out on the land presented its issues. While there was a lot more light, it doesn't get attenuated in the water, and there was certainly plenty of water to live on, the soil was nutrient deficient. Well, we think, and we have a lot of evidence to suggest this, that plants didn't move out onto land by themselves. They actually moved out onto land with a fungus that we'll learn more about in the fungus chapter called mycorrhizae. That's how you say this word, mycorrhizae. So we think that plants moved out on the land in a symbiotic relationship with mycorrhizae. And soon after they moved on the land, the plants, then animals followed. First was the arthropods. Arthropods are really good at living in relatively harsh conditions. Desiccation was a problem, and a lot of these plants moving out on the land had these waterproof coatings on their leaves. Even some plants today do, like red mangroves. Red mangroves have a very waxy coating on them. It's because they live in conditions that are very dehydrating and brackish water. And as I said, the plants created all these great opportunities for everybody else. Let me show you a mycorrhizae. And like I said, we're going to be looking at mycorrhizae more when we get into the fungus. But this is actually a fossil stem from an organism from, was it 405 million years ago? And what you can see is, this is like if they took a cross-section of the stem. All of these little black areas, they're called the arpusculal containing cells, that's actually a fungus. Now, sometimes when we think about a fungus attacking a plant, we think about it in the pathogenic sense, but this was in a symbiotic sense. You see, fungus go about consuming food in their environment very different from the way we do. We bring food into our bodies and we digest it inside of us. Fungus, mushrooms and such, digest on the outside. They actually produce enzymes that are then, just like we do, that are secreted to the outside environment where they dissolve the organic material that's there, and then they just basically drink up that dissolved material. So it's digesting the food on the outside, if you will. Now, mycorrhizae growing around the roots, all right, so again, this is a cross-section of the root. You're looking at the root of the stem, excuse me. The mycorrhizae growing around the outside, what they did is when they're digesting that material, when they're secreting those enzymes, they suck up some of the material for themselves, but there's always excess, right? It's like plants growing for photosynthesis. They make food for their own needs, and the rest they make for growth. And luckily, the herbivores can take advantage of that, right? So the mycorrhizae are digesting a lot more than they can use. So they're making this nice, already broken down, rich nutrient soup around the roots of the plant. So when the plants moved out on the land and the land was harsh in terms of the amount of nutrients, the mycorrhizae helped the plants deal with being able to absorb those nutrients from the environment. Now, this wasn't just something, these mycorrhizae weren't just something that was necessary back then. As a matter of fact, today, in today's world, almost all, 90% of the plants out there have mycorrhizae growing on the roots. So mycorrhizae have continued to this day to be an extremely important symbiote with plants. That's why you have to be careful sometimes if you're spraying an anti-fungicide on your plants, like if your plants get a fungal infection or something like that, you have to be careful spraying that, because if you get too much of it on the soil, it'll actually soak down in and kill the mycorrhizae, which in many cases will kill the plant, or at least stunt the plant. All right. Any questions on that so far? All right, this is a good place to talk about extinctions. Extinctions are a normal part of life on Earth. Things go extinct when they're caused by human beings. That's always traumatic, obviously. But as I said, they're a natural occurrence on Earth. The red line that you see in this graph is extinctions that have occurred, and we've identified 5 major ones with the red arrows you see down below. So there was an extinction way back in the Paleozoic about 40 million years ago. There was a really big one about 250 million years ago. We're going to talk about that one. This is the Permian period. So at the end of the Permian, there was an extinction. And then the other big extinction that we're going to discuss is the one that occurred about 65 million years ago at the end of the Cretaceous. And that's when the dinosaurs died off. The Mesozoic is the dinosaur era. So the Permian extinction heralded in the dinosaur era, and Cretaceous extinction, the second one at 65 million years ago, was the end of the Mesozoic era. A lot of times these eras start and end based on extinctions, as you can see here. The Paleozoic and the Mesozoic. The Mesozoic is separated from the Paleozoic and the Cenozoic by these two big extinctions. Let's talk about the Permian extinction first. Notice too, by the way, I do think it's interesting. Notice the number of families of animals keeps rising. So even through all of these extinctions, because oftentimes extinctions create opportunities for other organisms. So even though we've had some of these mass extinctions, the number of families of animals on Earth has actually increased. Okay. So about 250 million years ago, we have the Permian extinction. And during the Permian extinction, oh man, all hell broke loose. During the Permian extinction, most all marine species were extinguished, 90%, and about a third of all the insect orders were destroyed. This happened pretty quick. 5 million years. That's pretty fast in geological time. So the reason why that's significant is if you're thinking about adaptation, sometimes organisms will go extinct or they can't adapt because the change is just too fast. And 5 million years in the overall scheme of things is a relatively fast change. Well, what would have caused this massive extinction to occur? And there's two major ideas about what happened. The first one has to do with this continent maybe you all have heard of, the first major continent on Earth, which was Pangaea. And the other one has to do with these volcanic eruptions in Siberia that I'll get to in just a second. But let's take a look at this Pangaea thing again. I'm going to skip up a couple slides and then come back. Down here at the bottom, 250 million years ago, here we go, right there. As the Earth evolved through time, land began to appear, okay? Land began to form. And it was formed in little chunks, and as the chunks got bigger, eventually it formed this large, one single supercontinent known as Pangaea. That was 250 million years ago that Pangaea formed. Soon after that, Pangaea starts breaking apart. The continents begin drifting. Now, I know you guys are all familiar with continental drift. Continental drift has to do with the fact that the Earth is really it's a crust. The outside of the Earth is a crust that's broken up into all these plates that you see here that floats on this mantle. The mantle is melted rock, molten rock, okay? So these huge plates, these broken up pieces of the crust, they bump into one another. And as a matter of fact, if you look in the Atlantic part, you see the arrows, that's where parts of the plate are actually being produced. So at that, along what we call the north or the Atlantic, mid-Atlantic ridge, rock or crust is being formed, and as it forms, it moves away from that ridge, and it causes these plates to bump into one another. Sometimes plates dive below the other one, and this is where earthquakes and volcanoes, the little dots are earthquakes, and you can see a predominant number of earthquakes comes from areas around the plates. This is Wegener's theory. This is the guy who came up with it. Wegener's theory of continental drift. Now, Wegener, kind of like Lamarck, had the mechanism wrong as to why these plates shift around, but he did have the right idea. And what prompted him, and he wasn't the only one to see this, but was the interesting match, the jigsaw puzzle match between, like, say, you know, the east coast of South America and the west coast of Africa. There's other examples too. Mountain ranges. There are mountain ranges that start on one continent and pick up on another continent across the ocean. Like in South America, there'll be a mountain range that, you know, extends, say, this way across the continent, and then on the other side, yeah, the Ring of Fire. On the other side will be the rest of that mountain range. Well, the implication, or at least the assumption, might be that at one time those were all together as a mountain range when the continents were all together. Yeah, the Ring of Fire is all that around the Pacific. There's so much tectonic activity going on there. All right, so the plates are moving. The Atlantic is getting wider. The Pacific is getting narrower. So now we can go to that slide. So we start with Pangaea at the bottom, and we drift up 135 million years. We have Laurasia, which was the northern, and Gondwana. And then after that, right about the dinosaur era, the continents start to look very much like what we're used to today. Notice India is drifting through the Indian Ocean, and India actually slams into the Asian continent, pushes it up, and that's what actually causes the Himalayan mountains to form. Well, back to the Permian extinction. When Pangea came together, so we're down here at 250 million years ago, and that's when the Permian extinction happened. Here's what it did to ecosystems and the organisms living there. For one thing, the formation of Pangea caused the oceans to become deeper, right? And a lot of the inshore areas around the continents, around all these little pieces of continent before Pangea formed, were reduced. You started losing a lot of the area around the continents. Now, in the marine environment, most organisms and most productivity, or much productivity, is going to occur near the continents, right? That's where a lot of the nutrients are needed. That's where the water, that's where a lot of nutrients come off the continent into the water that are needed. That's where the water's shallow enough that you get photosynthesis, so you get producers. There's not nearly as much productivity out in the middle of the ocean. So most of the species, even today, folks, most of the species diversity and plentifulness, abundance, is going to be near the continents. Well, when Pangea formed, it reduced the amount of coastline. You know, if you imagine that there were a bunch of little tiny lumps of land, each one of those having coastline around it. Now, if all those little lumps of land eventually congealed together to this one supercontinent, you can do the math, you've lost square miles of coastline. And when you lose square miles of coastline, ecosystems suffer from that. So one of the reasons was this major depletion of marine species was the loss of habitat. You know, these continents formed, and now we lose that coastal area where a lot of this stuff lived. The formation of Pangea also made widely different areas in terms of climate. So along the coast, it would have been tropical wet, but in the very inside of the continents, the interior became a lot drier. So the inland climate become drier, become cooler, and the dryness was important because if you remember, we talked about this, amphibians have to lay their eggs in water. But reptiles and birds, for example, they don't have to lay their eggs in water. They have a shelled egg. So while amphibians probably would not have fared very well moving into the middle of Pangea where it's dry, where it's more desert-like, but there are other critters out there that would have done quite well, and that would have been the reptiles. You remember what I said, at the end of the Permian is the dinosaur era. All right, another thing that led, we think, to the Permian extinction was volcanic eruptions. And again, I want to impress upon you the magnitude of these volcanic eruptions. We're not talking about a volcano going off every once in a while. In the early Earth, it was basically nothing but volcanic eruptions. But during the Permian era, we have reason to believe, researchers showed us that at least in this one spot, in Siberia, I'm sorry this map is in Russian, it doesn't matter, you can still see where we are. The area outlined in the blue was a huge lava lake. So it wasn't like a volcano, but it was this, you can have thousands of kilometers across of nothing but this exposed, like a big gash in the crust of the ocean where all this molten materials at the surface. And it was giving off all these noxious gases, hydrogen sulfide and all that sort of stuff. So we had that kind of stuff being dumped into the atmosphere. And a lot of ash or a lot of material up in the atmosphere would have blocked out the sun and caused the temperatures to decrease. Yeah, heck yeah, it sounds horrifying. I mean, the scale of this, that's 1,000 kilometers. So we're talking, just real quick, 1,000. It's got to be 3,000 or 4,000 kilometers across. That's a massive amount of volcanic activity. All right, so after the Permian and this massive extinction and these formation of these continents and these diversification of habitats, right? You got places around the ocean, you got inland places. Then these guys came on the scene. Isn't that a great picture? I think about the dinosaurs as busting through the gate. Here come the dinosaurs. Now, they were small at the beginning and rose to prominence in the Jurassic, in the middle of the Mesozoic era. And they were doing quite well. Now, mammals were evolving during this time, but the dinosaurs were winning. They're a lot bigger and a lot meaner. And most of the mammals were basically night dwellers at that time. We know that because a lot of the mammal fossils we see, the mammals had really big eyes. So it usually means they're probably a night dweller. And these guys spent about 200 million years ruling the place. And they probably would have continued to rule the place had it not been for, I don't know, I guess for us, this fortuitous occurrence that happened one day. That's what I think is cool about this. It happened in one day, 65 million years ago, the Cretaceous extinction, when about half of the marine species went extinct, a lot of terrestrial plants and animals went extinct, and of course, the dinosaurs took a hike. Now, what caused the Cretaceous extinction was the subject of debate for many years until something called the Alvarez theory, named after the brother, no, it was a father and son, I believe, from Berkeley, who put this theory together. Their theory suggests that what caused the Cretaceous extinction was a massive asteroid strike. And folks, let me emphasize massive. This wasn't a little meteorite. The asteroid that struck the Earth that wiped out the dinosaurs was about the size of Manhattan. So this thing was huge. So it strikes the planet and throws up all this debris up into the air. And of course, it just caused all kinds of carnage. It peeled away, totally peeled away the crust of the Earth. And you know, like I say, it just was a horrible event on Earth to experience. One of the things that we were missing for many years, though, was the smoking gun, was the crater. Holy cow, an asteroid that big ought to leave a massive crater. We didn't find it, but lately, or as of late, we did locate what we believe was the crater. It's called the Chicxulub crater, and it actually is, can't really see it. It was a seismologist, people looking for oil and gas, that found it. They were doing seismic surveys, and in their seismic surveys, they found a big crater that's partially on land and partially in the water. So you can, now, you can see where we are in Florida. That would have really sucked, huh? We're living right here. And here comes this huge asteroid. Now, the Alvarez theory, where's their evidence? Well, if you look at the picture down below, what we've done here is scraped away the side of this rock, exposing rocks where the red arrow is, exposing rocks that are at the 65 million year old period there. And that dark line that you see there is a line it's very thin compared to the other rocks, but it's found all over the world. All over the world, where you dig down to rocks that are 65 million years old, you find this line, and it's an element. The line is made of an element known as iridium. Now, iridium is one of the periodic table of elements, and it's found on Earth, but very rare. It's very rare to find iridium on Earth. However, iridium is found in high concentrations in asteroids. So perhaps this layer of iridium that we see is the remnants of this exploding asteroid as it hit the Earth and threw up all this stuff up into the air, including parts of the Earth, but also the iridium from the asteroid. And that iridium then fell back down to Earth again and formed this layer. So that's pretty compelling evidence of an asteroid strike. And then, of course, after that, as I said, they found the crater. And for the most part, everybody accepts it that it was an asteroid. The dinosaurs might have been on their way out anyway. Conditions and environments were changing, but this was probably more like the nail, the last nail in the coffin kind of thing. All right, now we've already gone through this, so I'm not going to go through it again because we did it at the end of the previous lecture I did on this. But I do like this chart because it puts side by side the events that were occurring on Earth, amphibians, and when the first gymnosperms, and it also lays next to it what the continents were like at the time. So you can see just about the time the dinosaurs died off, the continents were almost in the same place they are now. So that's interesting to relate those two together because, of course, we know from the very beginning in our discussions of evolution that it's all about the environment, right? The environment causes organisms to adapt. And after that, after the dinosaurs went extinct, that opened up the gate for lots of other organisms. For example, the mammals, and again, 65 million years ago being just about right here, you can see how the diversity of mammals increased, especially among the eutherian mammals, marsupials. Monotremes are really weird mammals. They didn't change much. We're about 5 species. But that lifting of that heavy pressure of the dinosaurs and this diversification of habitats and diversification of ecosystems is directly evident in these adaptive radiations that we see. Now, we've talked about adaptive radiations before, species organisms speciating out and taking over a bunch of different environments. Now, it happens in animals. It also happens more regionally, like in plants, like on Hawaii. All of these plants that you see here, found on these different islands, were actually a close relative of a North American plant, the tarweed. And these, some ancestral tarweed made its way over to the Hawaiian Islands. And then throughout the years, you could see 5.1 million years or 1.3 million years, we get this adaptive radiation of all these different kind of plants from that. And that's what you oftentimes see, as I said, when a selective pressure is removed. You see this massive adaptive radiation. It's kind of like when you were a kid and in high school, maybe mom and dad were going out of town and they were going to leave the house to yourself. You know, holy cow, mom and dad are gone, but everything's going to go crazy. So the dinosaurs are gone, boom, everything radiates out. We got all these different species out there. Don't have to worry about those dinosaurs running around and trampling and eating us anymore. All right? Okay. Questions on that? Got a couple of things that I want to talk about genetically that may have happened or may have contributed to this rapid adaptive radiation, this rapid proliferation of species. Developmental genes. Developmental genes are genes that do exactly what they say. They control the development of an organism from when it's young until when it's larger. And developmental genes control things like how fast you grow, when that growth begins, or when that maturation process were to begin. You think, for example, like in puberty, that would be a developmental gene that controls that. And then other developmental genes control where things are, spatial patterns, where the legs are, where the petals of a flower are, where all these appendages, if you will, are located. Those are controlled by spatial genes. So if you make slight little changes at the very beginning, these developmental genes, and we're talking about through evolutionary time, if you monkey with these developmental genes just a little bit at the beginning, the little changes that you make at the beginning may end up yielding very profound results. Okay, so let's look at heterochrony. Heterochrony literally means different timing. And then we'll look at homeotic genes, and they have to do with the spatial pattern. So let's look at the rate and timing thing first. Here's what heterochrony basically says. We can actually see heterochrony in today's organisms. Take the skull of a chimpanzee fetus and the skull of a human fetus and put them side by side, and not too many people, except maybe a primatologist, would be able to tell the difference between the two. But now take the adult chimpanzee and adult human, and they're quite different. If you're following what they're trying to show you in the graph there, if we start off from the same place, the same basic shape, how do we end up with such dramatically different shapes once they become adults? Well, that's heterochrony. That's the rate and timing of genes. So the gene that would elongate the jaw, for example, in the human fetus, that gene would elongate the jaw to a point and stop. But in the chimpanzee fetus, as it grows, that gene would continue to elongate the jaw, so we end up with the chimps that have the much longer jaw. You could apply that to other parts of the skull. You know, the fact that the heavy brow of bone that we see in chimpanzees, the way the skull slopes towards the back, all of that could have simply been the same basic genes, but just controlling when they turn on, when they turn off, and how long they're on. Okay, so little tiny changes in the fetus part end up having very dramatic results in the adult part. It's kind of like, I think we talked in class a while back about how we could protect ourselves from an asteroid, and I said the best way is to just somehow slightly deflect it, like just by a couple of centimeters, you know, way out there in space. So by the time it gets to Earth, those couple of centimeters have added up, and the asteroid will miss it by quite a bit. It's kind of the same thing we see here. Now, heterochrony can also affect an organism and its ability to reproduce. Neoteny is referring to changes in timing of reproductive growth relative to somatic growth. Somatic growth is the growth of the part of your body that doesn't have to do with reproduction. Okay? So what we're saying here is if we change, basically, if we change when you become reproductively mature, we could actually get into a situation where we have, in the second one, paedomorphosis, we have an adult, okay, that actually looks like the juvenile of an ancestor. So we have a descendant adult, an organism that descended, through evolutionary time, natural selection, from another critter, but it looks like the ancestor looked like when it was a juvenile. And then if we get patagenesis out of that ancestral juvenile characteristically looking descendant adult can now reproduce. Right? I'll show you a couple of examples of what I mean. This big salamander here, Amblystoma mexicana, axolotl, right? Then right there, that's an adult. But among folks who look at amphibians and salamanders, that looks like a juvenile salamander. Juvenile salamanders have gills, and they look like they have that raised ridge along their back because they live an aquatic lifestyle. The slimy salamander, you see up above, we have those in Florida, very similar as well. Slimy salamanders and axolotls, we think probably went through a patagenesis event where basically they look like the juvenile, but now they're reproductively mature. And all of that, again, would have been controlled by rate and timing of those genes that control those processes. So you see what I mean? Little tiny, adjustments at the beginning can have very dramatic results at the end. Timings in the little adjustments in the timing of when you become reproductively mature could end up causing an organism to look like the juvenile and still be reproductively mature. Now, if you're thinking, why would that be an advantage? The quicker you start reproducing, the more organisms you leave behind, right? The earlier you start your life reproducing, depending on how long these organisms live on average, the more organisms you leave behind. And that's the basis behind evolution, right? How many critters you leave behind, how many offspring. All right, spatial genes. Hox genes are a class of genes we call homeotic genes. And a homeotic gene is a gene that basically, during embryonic development, tells the developing embryo where to put the appendages, where to put the legs, where to put the antenna, where to put the wings, where to put all that stuff. And some of the work that's been done has showed us that some of the modifications that might have occurred to these spatial genes that actually supported the evolution of organisms. So, for example, we're going to be looking at swimming versus feeding appendages in crustaceans. So the two crustaceans, or in arthropods. So what we're looking at is the Artemia on the right, which is a crustacean, and Drosophila, which is an insect. They're both arthropods. And they both have Hox genes. And they have two Hox genes. They have a UBX Hox gene, and they have an SCR Hox gene. Let's look at the UBX, the Hox gene number 7. If you look on the right with Artemia, lots of legs. Now, Artemia still is around today and has lots of legs, but we know one of the things that occurred during arthropod evolution was fewer and fewer legs. And not only fewer and fewer legs, but when we get into arthropods, we'll learn that arthropods are all about the appendages. It's a modification of those appendages. So the expression of that UBX gene, it does not suppress leg formation in crustaceans, while it does suppress leg formation in insects. All right, so we get an insect on the left from a crustacean on the right with lots of appendages to just a few. That might have occurred, we believe, looking back through the evidence, by a mutation. About 400 million years ago, we believe a mutation occurred among this UBX Hox gene, which actually took it from the fact that it does not suppress leg formation to now it does suppress leg formation. And it suppresses it differently throughout the body. You see in the Drosophila, the green little triangle up above, that means that the suppression of the legs is going to be less towards the back than it is toward the front. Now, this is all about spatial relationship of legs and appendages. So simple little genetic changes could have created a less legged organism from one that had more legs, right? And that would have allowed diversification as well. But when it comes right down to it again, and I've mentioned this a couple times, you've got to keep in mind that evolution does not have a goal. Now, I'm not going to sit here and talk about horse evolution. It's fascinating, of course, but horses evolved or started evolving from an ancestor, Hyracotherium ancestor, somewhere around 55 million years ago, and of course up to the modern horse. And all of these other species are other species that arose and went extinct. And some are grazers and some are browsers, and it all had to do with their environment. But I want you to understand that moving from Hyracotherium at the bottom to Equus up at the top wasn't a direct route. Evolution is like a wandering sort of path, back and forth. These species arise and fall, and these species arise and fall. So it wasn't like there was one species that was destined to be the pinnacle, the Equus. All of this is trying out, right? All depending on the environment. No goal in evolution. All right, any questions on that so far? One more thing I need to talk about with you guys on this. Eyes, feathers, legs even are fairly complex structures. Matter of fact, we call them evolutionary novelties. Evolutionary novelties means a really sort of interesting evolutionary structure that arose. Stephen Jay Gould, a modern evolutionary biologist, used to write an article, and one of the quotes in there he writes is, "What good is 5% of an eye?" We could say, what good is 5% of an eye, legs, or feathers? Now, the 5% is just a rhetorical number. What he's basically saying is eyes are pretty complex structures, and they need all the parts there to work. It's really not very parsimonious to think that all the parts of an eye evolved together to make the eye, right? The modern eye that you have today, that really complex structure, it's hard to believe that that all evolved, all at one time. So this explanation of these evolutionary novelties involves something we call acceptations. That's the word down below. And acceptations means that a structure evolves initially for one particular purpose, one particular survival purpose, that would be the evolutionary part, but ends up becoming fortuitous in another context. All right, let me show you what I mean by that first. Let's look at the evolution of eyes. So we're going from the evolution of eyes from A all the way down to E. We're looking at a limpet. Then there's a mollusk. Nautilus is another mollusk. A lot of mollusks here. And then finally, the squid is a mollusk as well, too. So here's what we think. Now, these organisms exist today that are listed in this screen, but, they represent an evolutionary past in some cases. A patella or limpet has does not have an eye. It's got a series of pigmented cells, photoreceptors. It can sense light. So you can imagine if a predator were to swim over a limpet, it wouldn't know what predator it was, but it would just sense the shadow. And if it had an evolutionary advantage, well, then that means that that organism would, when it would sense that shadow, would go off and maybe hide. Right? Doesn't know that that was the predator, but why not err on the side of caution? Now that would be selected for, wouldn't it? Protecting yourself against predation. Next, what we think might have happened is these pigmented cells began forming into a cup, an eye cup. And what an eye cup would do is it not only gets a little better at focusing the light, but it also lets you sense light from different directions. See, this here on the left, you can only really sense the light from the front. So now you get a sense of which way the light's coming from. And then as we move through the nautilus and the murex and the squid eventually, what we're doing is we add a fluid-filled cavity. That's going to help with resolution. That's going to allow you to resolve the image more than just a shadow. Adding a lens is going to help you with resolution. And then if you can control that lens, that camera lens type, you can focus it. So the eye down there in the loligo, the squid, didn't pop onto the scene just like that, right? It was a series of these little changes that lended an evolutionary advantage all the way along the way. That's why they were fortuitous in the final context, right? These were structures that were initially set up to sense light. Now, we're going to look at some flatworms later on in the semester, and they have eye spots. They have eye spots. They don't have eyes. They have patches of pigmented cells. So the creatures that are still around today that have that. So that's what Stephen Jay Gould was saying. We didn't start with 5% of an eye. Each thing along the way conveyed an advantage to the organism, being able to tell shadow, being able to tell where the light came from, being able to focus and resolve whatever that shadow was, and eventually being able to see it, kind of the way we do. Squid are interesting critters. They have eyes very much like ours. So again, and kind of just using a different set of pictures, an early chordate, an early ancestral vertebrate might have had an eye spot, and then some creature may have come around that actually had these light sensing cells and bulged out from the side. So that means that they could see, in directions. They could see out here and see out there. There are even some deep sea creatures that are around today, some species of shrimp that have a retina that curves, and they actually sense light coming in. Oh, I drew that the wrong way. It's like this. Well, hold on. It's like this. So they can sense light coming in from the side, so they can see to their side and above them at the same time, so they can watch for predators. So maybe this bulging out would have helped with that. And then like in the hagfish, you start to see the cup, right? And initially the cup is covered with a lens placoid. And a placoid is just a collection of dermal tissue. And that lens becomes more and more transparent with time. It becomes more sophisticated. The lens now has the ability to focus. And then eventually we get to the shark, where we have this much more sophisticated eye with the cornea and the iris and the focusable lens. So an eye didn't pop onto the scene like it did with the shark right here. Eyes evolved slowly through time without any goal in mind. The goal from the pigmented cells in the eye patch, or the eye spot right there, like in A, the goal wasn't to make a sophisticated eye. The goal was simply to allow the organism some way to sense its environment, which would increase its survivability. And through time, that was built upon. So it was part of an eye at the beginning, but it was still a functional object at the beginning. I guess I'm wanting you to understand that you don't create an eye by making parts of the eye through time, and then all of a sudden you got an eye. There's no way Mother Nature can predict the future that way. Each structure, each eye-like thing, okay, eye cup or pinhole camera type eye or whatever you want to call them, has to convey an advantage to the organism, that ability to sense light, right? Now, we'll talk more about this when we get into feathers. Feathers in birds didn't evolve to help feathers or help birds fly. They evolved for a different reason. And legs, a similar sort of thing as well. Legs didn't evolve to help organisms walk on land. There was another reason for it. Okay? All right, folks, I'm going to stop the recording.